Okay, welcome everyone. It's my pleasure to welcome you again today to a joint EV GUSEV club, which is co-hosted by the special interest group on genitourinary system EVs. We have a great talk prepared for you today, presented by Gloria Ravegnini. Gloria is a senior assistant professor from the Department of Pharmacy and Biotechnology at the University of Bologna, where she explores the mechanisms of cancer development using molecular biology. And she is going to talk to us about the distinctive features of blood and ascitic fluid derived extracellular vesicles in ovarian cancer patients. And before I give her the floor, I just want to tell you a few words about the questions afterwards. So if you have questions, please post them in the chat space. And depending on how crowded it gets, I will either let you unmute or give the read the questions out, and then Gloria can answer. If we run out of time, of course, I think it would be nice to send every question to Gloria, and she can maybe approach you personally. So with that said, Gloria, thank you for joining us today, and the floor is yours.
Thanks, Elena, and thank you for this invitation. I'm very glad to present our recent work that, as you can see by the title, is focused on ovarian cancer patients. And in this work, we analyzed matched material, body fluids, blood and ascitic fluid from ovarian cancer patients. And I will explain you in a bit why we decided to analyze these two different fluids. So first of all, I will give you a brief introduction on the ovarian cancer patient for people not very familiar with this disease. So ovarian cancer patient is a tumor that actually is considered not so common. We have about 200,000 new diagnoses every year at global level. In Italy, we have about 2,500, 3,000 new diagnoses. But beside this low considered incidence, this tumor is actually a big killer. Is also known as a silent killer, mainly due to the fact that there are no diagnostic biomarkers for the early stage of this disease, and there are no specific symptoms. So the result is that the majority of cases are diagnosed in an advanced stage, when the chance to get a complete cure are more limited. Consider that patient diagnosed in an advanced stage have a 5-year survival rate of about 10 to 30%, while when this disease is diagnosed in the early stage, the 5-year survival rate is about 80-90%. Beside this high lethality, ovarian cancer is also characterized by chemotherapy resistance that usually appears within the first 2 years after the start of the treatment, and very important, is characterized by a wide metastatic spread. And in ovarian cancer, the route for this dissemination is not the much more common lymphatic or blood route, but rather the transcellular dissemination. What are these two different types of dissemination? So basically, in the majority of cases, we know that for having metastases, we need that cancer cells leave the primary site, reach the bloodstream of the lymphatic route, and then can engraftment in distal place. In ovarian cancer, however, this is not the main route. Indeed, the metastatic dissemination happens mainly through the abdomen cavity. So in ovarian cancer, in particular, often in most of the patients, we have accumulation of ascites. So in physiological condition, every of us has ascitic fluid in amount of about 1 or 2 ml. In ovarian cancer patient, this liquid can reach up to 1 or 2 liters, of fluids. These, of course, cause very discomfort, much discomfort for the patient and poor quality of life. Ascites is also responsible in particular for this dissemination. This liquid completely covers the tumor, and the tumor can freely release in this fluid different amount of circulating molecules, ctDNA, ctRNA, circulating tumor cells that in this fluid are often found as big cluster, and extracellular vesicles. The exact mechanism through which the dissemination and the start of metastasis spread arise is not very characterized. One hypothesis is the so-called seed and soil theory, according to which the extracellular vesicles are able to reach the distal site, transfer their content, and prepare the soil for the engraftment of the circulating tumor cells in a second moment. So when we start to evaluate the extracellular vesicles in the ascites in ovarian cancer patient, actually there were limited studies and there were no comparison between the characteristic release in ascites and in blood derived in blood in this patient. So we suppose that actually the population of extracellular vesicles retrieving blood and ascites had to be different and had to have different function. So for this reason, we aim to characterize the future of extracellular vesicles released in blood and the ascites of ovarian cancer patient in terms of the dimension, in terms of the expression of superficial protein, and in terms of content of microRNA. So in a collaboration with the Sant'Orsola Malpighi Hospital of Bologna, which is a reference center for gynecological cancer, we started to collect material from ovarian cancer patients. The university, the hospital, the Sant'Orsola Malpighi Hospital has about 500, 600 surgery of gynecological cancer per year. So we had the opportunity to collect material from this type of tumors. And we collected for each patient the blood, the ascites, tumor material, and normal material. These patients were included in the study before the start of the treatment at the time of the diagnostic laparoscopy. And we included in total 60 patients. As I mentioned, we characterized the extracellular vesicle for the two body fluids in terms of size and expression of the superficial epitopes. And for these, we isolate the AV using a specific column from the ISON. While for the characterization of the microRNA, we use a kit from QIAGEN that is specific for extracellular vesicles. Currently, we are also collecting urine and saliva, but at the time we did not have this fluid to include this additional biological fluid and compare eventually the extracellular vesicles. So regarding the dimension, we employ nanoparticle tracking analysis, and we analyzed 37 patients for a total of 74 samples. We observed a difference in terms of dimension for both the mean and the mode, and I mean a different significance from a statistical point of view, observing that in ascites we had larger vesicles. We also looked to the vesicle concentration. In this case, we did not observe statistical significance, but in serum, there were a little more vesicles, particles for ml. We also checked the dimension with the transmission electron microscopy, confirming the different dimension in the two fluids. The different sizes so suggested that probably these extracellular vesicles could be different also in terms of expression of epitopes. So we used specific assay by Milteni Biotech. This assay is a based multiplex flow cytometry assay, which employ dye label capture beads and allow to analyze 39 different protein with 2 protein as a control, so 2 control epitopes. And in this case, comparing 30 patients that are 60 samples, we observe 20 epitopes that were differently expressed in the 2 biological matrices. This is just an heat map to summarize the result that we got. Actually, we didn't expect to obtain this amount of difference. This just as example is a figure that represents the different expression of our epitopes. It was a bit difficult to interpret these results, so we looked in literature to understand if some of those have been already reported for ovarian cancer, and the most significant evidence that we found are summarized in this table, where the red blot indicate an important, a strong involvement in the metastasis process. Yellow dots indicate a mean involvement, and green low involvement in the metastatic process. Among the top most deregulated, CD44 and CD24, as you can see, were also reported as strongly involved in metastatic process due to the involvement for formation of spheroid and the involvement in the epithelial mesenchymal transition. And we were also particularly interested in EpCAM, CD326, that actually was one of the top most deregulated between ascites and blood serums. And this to us was also particularly interesting because as you know, EpCAM is also a marker to select circulating tumor cells. So the fact that the EV in extracellular vesicles also strongly expressed EpCAM to us was of particular interest. As a last part of our analysis, we evaluate the microRNA biocargo for these extracellular vesicles. As I mentioned before, we isolate the total RNA with a specific kit from QIAGEN, and then we use this array from Thermo Fisher that allow to evaluate 384 microRNA per samples, including 5 internal control. We then run this array, and then after we got the result, we evaluate the result performing principal component analysis, evaluating the differential expression of microRNA between our biological matrices, pathway-based enrichment, and target gene prediction. So for this profiling, we analyze 60 samples from 15 patients, matched samples of 15 patients for whom we analyze ascites, blood serum, tumor samples, and normal samples. And these are the results that we got. On the left, you can see the principal component analysis, where you can recognize the 4 different groups. On the lower part of the principal component analysis, you can see the expression of microRNA in vesicles released in ascites or in blood serum, and in the upper part are the tumor material and the normal material. On the right, you can basically see the same type of clustering. This is an unsupervised clustering heat map, where you can recognize the different pattern of expression that are associated with the different type of samples. Giving the time, I will focus just only the comparison between blood serum and ascites. Basically, comparing these two types of fluids, we retrieved 98 microRNA that were deregulated with a p-value adjusted lower than 0.01. For the selection of microRNA, we took in consideration the AM score. So the AM score is a score that probably many of us are not familiar with. Is a score that is released by the software that we use to extrapolate the result by the instrument. The company, the Thermo Fisher, which is the company that produced the array, suggests to consider the AM score when you have to select the microRNA to be validated. This score is basically a score that gives you an idea of the goodness of the amplification of the amplification curve of the specific microRNA, and they suggest to take in consideration an AM score higher than 1 in at least 50% of the sample. So in our experience, when we have to select microRNA to be validated in single assay, we know that considering the AM score give us the highest chance to confirm the result that we observe in the profiling. Of course, of 59 microRNA, we could not validate all of them. So we selected 9 microRNA to be validated based on the top score, deregulated miRNA, and so taking in consideration the evidence in literature regarding the ovarian cancer. Those are the 9 microRNA that we selected to be validated, and the validation was performed in 120 samples from 60 patients. The majority of these microRNA actually are from the family of microRNA 200. This is a family of microRNA that is actually reported as very important in ovarian cancer. And all the microRNA that we selected were actually confirmed at the for the statistical significance level. And actually some of them were also more significant after the validation in this extended cohort. We evaluate a functional enrichment to understand the pathway in which these microRNA were involved. And as you can see here, most of the pathway, the function of these microRNA are associated with the epithelial mesenchymal transition and the mesenchymal epithelial transition. So when we submitted the first time the paper, we did not include any type of validation, functional validation in cell line, but then 2 reviewers asked to include some validation regarding the association with this epithelial mesenchymal transition. So we perform a mimic transfection for microRNA 429, 21, and the 200 A, B, and C in SCOV3 and OVCAR3, that are 2 ovarian cancer cell line. We confirm the increase of microRNA by real time, and these are the results regarding the modulation of some gene involved in the epithelial mesenchymal transition. Even if as I mentioned, we were more interested in characterizing just the DAV rather than in this paper including also the functional validation, because they're still in process. But as you can see by this preliminary result, most of the mimic that we used actually seem to modulate genes involved in the epithelial mesenchymal transition. We also evaluate the association with clinical parameter. Interesting, we observe that the significance with the progression-free survival and the overall survival were just for potential biomarker in AV release in ascites. So regarding EPCAM, we observed that patient with higher expression on EV released in ascites had shorter progression-free survival and shorter overall survival. And so for microRNA 1A1, just in extracellular vesicles released in ascites, patient with higher expression were also the patient with shorter progression-free survival and overall survival. We also evaluated the association with two biomarkers that are used in ovarian cancer, CA125 and 19.9. In this case, we observed that these two biomarkers at the diagnosis were statistically associated with the microRNA 200 A, B, and C that was actually associated just with the CA125, while microRNA 429 was associated with both CA125 and CA19.9. So concluding, this part regarding the paper, in this first work, our first work on extracellular vesicles, comparing the two fluids, ascites and blood serum, we observed that actually these extracellular vesicles are different, are released in a specific way in blood serum or in ascites, and it seems that the ones released in ascites have future that could be promote the metastatic potential in this cancer patient. And ascites seems also to be a very rich source of tumor-derived EV that could be also used as potential biomarker for peritoneal metastases. At the time when we started to work, we found a similar result in patients with pancreatitis, in which they have ascites, and the tumor associated with the hepatic tumor, I mean the EV released in the blood, were different. But in the ovarian cancer, we did not find any similar studies. So this actually was the first study that tried to compare this type of extracellular vesicle in the two body fluid. So what we would like to do now? So we are trying, we would like actually to load the extracellular vesicle or exosome released by ovarian cancer cells with specific microRNA to try to better mimic the physiological condition. So preliminary, in collaboration with the University of Pisa that has wide expertise in this, we try to characterize the release of extracellular vesicle exosome in SCOV3 and in Ovicar. So we, as you can see by the workflow, we isolate in this case through sequence ultracentrifugation and then with the size exclusion chromatography. We isolate the extracellular vesicles. We check for specific EV markers in Western blot, as you can see here, CD81 and the TSG. We got positive signal for both the exosome derived by SCOV and Ovicar. And then we try to characterize the particles number also, and also the size. This was done easily for the SCOV3, as you can see by the tables, but we had some issue for Ovicar. This probably is due to the fact that as you probably know, when you isolate the exosome or the extracellular vesicles from cell line, you need to perform starvation for 24-48 hours. And we had a lot of issue with the Ovicar 3 because even after 24 hours, cells tend to detach, and so it seems that the sample is quite damaged, or there are many aggregates. So we are not able so far to measure the exosome released by this cell line, but given the yield from SCOV3, we think that after the vacation, we will try to load some microRNA in exosome from SCOV3, and probably we will start with the microRNA 429 that actually was, it seems, the microRNA with a bit more consistency in the experiment with transfection mimics that we done. So if any of you has any suggestion for Ovicar, are very welcome. I thank you, all of you, for your attention, the organizer for this invitation, and of course, all my group, in particular, Dr. Gorini, that had me with the bunch of experiments that we perform, and so the Policlinico Sant'Orsola Malpighi Hospital with Dr. Perrone and Jaco, because are the very important person that help us in collecting the patient material.
Thanks, Associate Professor Ravegnini, for your wonderful presentation. It's very interesting to see how you've looked at the extracellular vesicles from both acidic fluid and blood and how they compare. We'll now open the floor to questions, and there's already a few questions in the chat. So the first one is, there are three main cancers that have this behavior: serous ovarian cancer, adeno-ovarian cancer, and primary peritoneal cancer. Did you find any difference?
Yeah, thank you. Yeah, thank you for this question. I didn't specify, but those are all serous ovarian cancer, high-grade serous ovarian cancer. We did not include other additional types because we want to limit the bias for this part.
And the next question is, do you know whether patients with these metastatic cancers from such microRNA containing EVs in their circulation? If so, are they characteristic for the different cancers? This question refers to the previous question, whether there were different cancers. So we'll move on to the next question then. What is the function of the highlighted microRNAs per the tumor versus host response?
Yeah, actually, the function of the...
So I had two questions. Why did you pick the microRNAs of all the different RNAs you could have? What was the percent of the microRNA total that the highlighted ones were? And what were the fraction of the total RNAs represented by the microRNAs? And of course, the long distances, what could be the function of these microRNAs for the tumor versus the host response?
Regarding the fraction of microRNA isolated from the total, actually the kit from QIAGEN give you the opportunity to isolate the total RNA. Then we perform retrotranscription for microRNA with a specific kit from Thermo Fisher. So actually, I don't know the fraction of microRNA and the total RNA. We usually have a very good yield for when we retrotranscribe microRNA, but according to the protocol, we don't measure the concentration because we work in volume. And so the protocol is very specific for this part, and the only way that we can normalize is using the internal control for each sample. So we actually don't know the percentage of microRNA isolated with the kit from QIAGEN.
Well, it's very controversial whether the microRNAs exist in exosomes, EVs, and what their significance is. So why did you focus on the microRNAs versus all the other different kinds of RNA?
Yeah, we would like to, of course, evaluate the total RNA. I would like to perform RNA-seq, for instance. But when we started, we were not sure to have enough material from the extracellular vesicle. So we started from microRNA that for this type of profile require lower amount of material. But for sure, I would like to perform RNA-seq or at least a messenger RNA sequencing, for sure.
I agree with you.
Yeah,
that they're very important.
Yeah, and actually, we are trying to collecting matched material from the extracellular vesicle and the circulating tumor cells, because I would like to evaluate what the content of both. But for sure, I agree with you. We started from the easiest, from the easiest part.
Of course. Thank you so much.
Sure.
Thank you. Frederick?
Yes.
You can go. Please.
Yes, I'm here.
So you had additional question as well?
I can't see the question. Wait a second, I have to see my question.
I think that is in the chat. Can you imagine ways to use this information for screening?
No, it was about whether the exosomes get into the circulation and whether there's a characteristic pattern that that you could find in exosomes in the circulation that would indicate that it had come from metastatic ovarian cancer.
Yeah, this is a great question, because actually there are no way to understand if real the exosome reach in the circulation derive from the tumor. So the only things that we could do is maybe try to search if there is inside some, for example, I don't know, RNA that messenger RNA or protein that is strictly correlated with that tumor. But isolate the exosome from the circulation and understanding the percentage of that I'm sure there are strictly derived from the tumor is a very odd question, because we also try to discuss this part with the oncologist, but so far we didn't get any conclusion. Of course, some amount of exosome or extracellular vesicle from the tumor, I'm sure that will reach the circulation. But yeah, we are open to suggestion, because this is very important. And actually, I don't know if we could do that, but beside what I told before, to analyze the CTC and AV release in extracellular in ascites, would be very interesting to do the same things in circulation. So the most critical point for this part is that the circulating tumor cells released in the systemic circulation are very limited. We talk about 2 CTC, 3 CTC per every 5 ml of blood. We did preliminary evaluation for both the blood and the ascites. In the ascites, when we try to measure to perform a measurement of the number of the CTC, we saturate the signal of the instrument, because we have tons of circulating tumor cells in the blood. In 5 to 7 ml of blood, we usually have 1-2 circulating tumor cells. So in that case, the most difficult part is having enough material to perform this type of analysis. So yeah, this is what I would like to do, but I don't know if we will able to do that.
We have such a protein, not an miRNA, that we are looking for in the circulation from ovarian cancers. If you're interested, we could speak.
Sure. You work on ovarian cancer too?
I'm sorry?
You work on ovarian cancer?
Yes.
Yeah, for sure. Thank you. I will sign your contact. Thank you so much.
If I can go to my third question, it's relevant to the other one. Serous carcinoma, the one you looked at, is a droplet cancer. It starts in the fallopian tube, and then you have droplets of cancer falling on the ovary, and and then it embeds in the ovary, so it's a metastatic cancer of the ovary. And I think Phil's questions are particularly relevant because someone has to try to understand how the droplets embed in the ovary and particularly why they don't embed, and therefore one could perhaps think of a way to frustrate serous carcinomas from happening. Presently, the way to deal with them is to remove the fallopian tubes in prophylaxis, as I'm sure you know.
Yeah. Thank you for this comment.
I think that's a very relevant question in terms of metastatic potential metastatic spread mechanisms. Actually, it's really interesting to study if EVs actually prep the niche for metastasis in there. I also see a question from Thakur. Would you like to unmute or shall I read the question?
Hi.
Hi.
Yeah, so my first question was related to the 384 miRNAs that were checked from the Thermo Fisher design. These are targeted mRNAs. What was the approach while deciding these 384 miRNAs?
You mean why we decided to select this type
Yes.
of yeah, we actually work from a lot of time with this type of array. So we had wide expertise from this type of array. I agree with you that probably an approach with Illumina would be better. In the last year, we actually started to prefer Illumina because give you the global expression of microRNA. Yeah, at the time when we started the study, we didn't have that expertise and the instrument that we acquired later. And this is the easier response. But I agree with you, and we are doing this second approach in the study that we started recently, because for a profiling, of course, the global profiling that give you Illumina is the best approach.
So my second question was related to like if there is any treatment profile of the patients, will the expression of these miRNAs change based upon the treatment profile?
Okay, so these samples were all collected before the start of the treatment. So we didn't include any treated patient to limit, of course, the potential bias. But with this patient, we are so analyzing. We actually analyzed the profile of microRNA. This is not included in the present paper. But we checked the different profile after the first 3 cycles of chemotherapy, because those patients, as standard approach, are treated with chemotherapy based on cisplatin and Taxol. And there are, of course, difference. We have many microRNA that change after the first 3 chemotherapy. We also try to look if after 3 cycle of chemotherapy, the change of this microRNA can be associated with the response, because we know that most of these patients can show resistance within the first 2 years. So we are collecting the results because we don't have, from a statistical point of view, enough events to perform any correlation. So we need to wait a bit more to have a longer follow-up. From what we saw so far, there are changes in the microRNA that are statistically significant.
So I have last question. Is it related to only the ovarian cancer, or these miRNAs are also found in some other type of cancers?
No, those are, I mean, for example, the microRNA 200 family is very important in ovarian cancer. And this, for example, is one of the cluster of microRNA that change with the chemotherapy. But those microRNA are reported so in other cancer types. So are already reported, some of those as very important in cancer in general. In ovarian cancer are also already reported, but as I mentioned before, we were trying to be more focused on the changes between the 2 different fluids. So we didn't analyze, for example, this microRNA versus patient with no cancer. So they are deregulated in the comparison that we perform, and they are statistically significant, but we know that are already reported in ovarian cancer patients, comparing, for example, with non-cancer patient, or they are statistically highly expressed in tumor material compared with non-tumor material, and they are not new, we could say.
So I just asked because if these miRNAs can be considered as a biomarker for the health status, like with the chemo, if their expression is improving, can they tell us, okay, this is improving in the patient, that's why their expression is changed? So if it is present all over in the other cancers also, so is there any specific that we can find out?
For sure, for example, the 200 family have increased level associated with the stage of ovarian cancer patients. So in healthy material, for example, the expression is low and increase moving from the stage. I didn't actually remember the direction, I mean, the upregulation or the downregulation in the chemotherapy response, but if you are interested, we can for sure discuss about that.
Oh, thank you so much. Thank you, I appreciate it.
So I think I go next, is that correct?
Few I see.
Yes.
Again, ESO sites better than serum for potential markers
I
diagnosis.
can talk for myself.
Oh, sorry.
Yeah, is the ascites better than serum for markers? And it seems like maybe 24 and 133 are more associated with the host response, and the 200s with the tumor. But I don't know much. This is fantastic work to have, Gloria.
If you want, I will be very happy to discuss about that, because actually I think that was not so well understood by by the reviewer, for example. So maybe they were not very familiar with ovarian cancer, because when we discussed this data with our oncologists, they were super interested, no, in this work. So I'm happy that you appreciate the work.
Very important. It's a very difficult cancer. This is all new data. And as you see, I'm interested in what is the host response, microRNAs,
yeah,
versus what are the tumor.
didn't. Yeah, we didn't go too much through this part, because it's quite difficult to understand, no? And there were no data, so it's always difficult also to find people to discuss the data in order to better understand. So
I would be glad to discuss the data with you anytime you'd like.
Oh, I appreciate it. Thank you so much. I will be in contact with you.
Thank you.
So there is another question here. Actually, I have a question which is kind of related. So if you are to transition this to a diagnostic test, what biofluid would you be going for? And that's also related to the question from Emma, because she asked why choose for serum instead of plasma.
Okay, the difference, I would like to evaluate the difference between serum and plasma, but we started to collect both the fluids because there are reports in literature that, for example, serum and plasma contain different amount of protein, and there is not so much about the extracellular vesicles, but I think that would be interesting in understanding this part. The response for this question is very easy, because when we started to collect the blood, we asked to the nursery to collect plasma. Okay, but they started to take the wrong tube. So they collected the first samples in the tube with the serums, the serum separator. And so after that, we had the 10 sample like that. We decided to go with the serum. Yes, easier and honest response.
Yeah, typical. And if you want to move this to, if you want to move this to the diagnostic, to the developing of a diagnostic assay or test, would you be staying with the serum, or would you consider other biofluid as well?
Yeah, no, we actually for example in endometrial cancer we are using plasma. So I'm honestly, I don't know if there is a difference in terms of extracellular vesicles, and but yeah.
Okay. There's a question from Frederick again. Actually, is that differences in EV from chemoresistance versus responsive ovarian cancer? So can you use this prognostic biomarker or predictive maybe even?
In the so far we have the material to perform this, but we didn't use the characterization of EV, but is our intention to do that. So far we did this just for microRNA that as I mentioned, there is different, but we still lack the part regarding the expression profile of epitopes and the dimension.
And then the last two questions from Amy from Tekur. Did you deplete blood cell EVs from your EV sample before FACS? Because platelet-derived EVs can also contaminate the sample.
No, we didn't do this part before the analysis with the cytopherometric analysis.
Okay. Well, I think if there are no other questions or comments, I would like to thank Gloria again for joining us today.
Thank you. It was a very pleasure, and I'm happy of this question. So I will in touch with Friedrich and Philips, I guess.
Yes.
And I appreciate so much your comment.
And thank you again for joining from all over the world, also on impossible times. I think I'll close with that. Thank you again for joining, everyone. It was a very nice presentations with a very lovely discussion. Thank you.
Thank you so much. Bye.
Bye.